Imagine a woman giving birth to twins. They share a mother, a womb, and a birthday, but they do not share a father. Each baby has an entirely different biological dad. Your first instinct is probably to call that impossible. It sounds like an internet myth or a plotline ripped straight from daytime television. We are taught a very specific set of rules in high school biology. One act of intercourse, one egg, one sperm. The math is supposed to be clean and simple. But this phenomenon is real. It is a documented medical reality called heteropaternal superfecundation. To see how this happens, we have to throw out those simplified textbook generalizations. We need to look closely at the highly precise and surprisingly flexible timeline of human reproduction. Usually, the ovaries release a single egg, but sometimes a hormone shift or fertility treatments cause hyperovulation, the simultaneous release of 2 eggs. This double release is the standard requirement for creating normal fraternal twins. The presence of 2 viable eggs opens a unique biological window. The reproductive environment shifts from a single potential pregnancy to 2 parallel, independent possibilities. Now we have to look at the male contribution, specifically the viability of the sperm. Sperm can survive inside the female reproductive tract for 5 days. On day 1, partner A's sperm enters. On day 3, partner B's sperm enters, mixing with A's living cells. The eggs drop, and each is fertilized by a different partner. 2 distinct eggs, 2 different fathers, just 1 pregnancy. Perfectly normal biological variables overlapping on a specific timeline to create a highly unusual reproductive outcome. Fast forward to the aftermath. When these twins are born, genetically speaking, they are not standard twins. They are half-siblings who happen to share the same womb for 9 months. Because they draw from 2 distinct paternal genetic pools, the children might look entirely different from one another. Their eye color, their hair texture, and facial structure could be completely distinct. Or they could inherit enough dominant features from their shared mother that they simply look like regular fraternal twins. Without a stark visual contrast, nobody suspects a thing. These DNA sequence readouts show exactly how this is ultimately uncovered. Without obvious physical clues, the phenomenon is almost exclusively discovered after the fact, strictly through DNA paternity testing when doubts arise. This leads to an interesting statistical reality. Most standard twin pregnancies are never subjected to dual paternity testing. The real rate of this biological event is likely happening quietly and far more often than recorded medical data suggests. The human body rarely follows a rigid script. It operates within a layered landscape of complex possibilities. Science has a way of turning what sounds impossible into something entirely logical once you break down the mechanics. This is Anatomy Corner. Subscribe for more honest, jargon-free biological breakdowns, and you will never look at the human body the same way again.